I don't know why I have my mask on. Hey, everybody. So we have a few guests in the lab running gels. So if you hear, cursing, or if Jared starts to sob, it's because he misloaded his DNA. And so partly in response to Alyssa and Hannah coming by, and what I kind of promised was to go over this a little bit. It was meant to be an introduction to BLAST. And we've been talking about DNA sequence homologies and the ability to use a DNA sequence or a gene from one organism to pull out the homolog in the other. And this is sort of demonstrating this practicality. And so BLAST has changed. They've been threatening to change for a year or so. Over the summer, they completely did shift it. As I said, this is somewhat written for the old BLAST, but I kind of modified it a bit. But there's still a few things in here that, yes, I have to go through and remodify because the new graphical interface is a little bit different. Regardless, let's look at some of what we're asking to do. And there's a couple of things that I'll just tell you a little bit about. So, the idea about the amino acid conservation and things is part of what we're looking at, in that, let me see if I can view this in Google Docs. I'm just going to view this where it is. So, the first alignment that we're doing is actin, looking at flies versus humans. And I think that you'll find when you do the BLAST that these two genes are really conserved. As it turns out, fly actin will absolutely work in humans. And so I'm bidding farewell to one of our visitors. Farewell, visitor. And that's, I think, in part is because evolutionarily, the fact that actin interacts with so many other proteins, if it changes too much, then all those other proteins need to change, and they're interacting with a bunch of stuff. And so, really, actin is fairly evolutionarily constrained in terms of mutagenesis, or in terms of single nucleotide polymorphisms resulting in amino acids. And one thing to say is that we're looking at the amino acid sequence. If we're looking at the nucleotide sequence, it would actually be a bit more variable, but the amino acid sequence, it is really homologous. So, the different tabs that I directed you to, the plus sign in the middle row indicates that it is a conserved amino acid. And use a marker and marking the differences in the residues, and you can decide whether they are conservative or non-conservative looking at the handout. This actually is not the version I sent you. I edited it. This is off my Google Drive. But I sent you that little block from Wikipedia about the conserved amino acids. So actin is going to be really highly conserved. Next, we come down to Huntington. This is the gene product that is the causal mutation in the neurodegenerative disease, Huntington's disease. And Huntington's disease is a dominant mutation, and it results from what's known as a polyglutamine repeat. And actually, if you look at the sequence of it, it's pretty darn easy to see the polyglutamine repeat. Like right there is a bunch of Q's. And I'm going to highlight that. Oh well. Anyway, you see, that's the polyglutamine repeat. And this repeat sequence is actually what generates the deleterious effects in the protein. Huntington's has a neurodebilitative consequence that, depending on how many Q's you have, comes on earlier the more you have the repeats. And if you have a few outside of the normal range, it comes on later. So Huntington's is not as well conserved as actin. There are a lot of different differences even between two vertebrates, fish and humans, which are fairly evolutionary separate, but they're still vertebrates. And the gap in the alignment and the sequence from one species has an insertion relative to the sequence of the other. Which one has it? See if you can find that. How might it arise? Well, you can look in your book. You can actually sort of think about this. We'll be talking about some of these sort of genomic rearrangements and things that occurred in a bit, another couple of weeks, in terms of genome evolutions and things. All right, again, highlight the amino acid changes. And the conservative substitutions, mark them with a plus. They should actually have a plus with them these days. Back in the old days, they didn't always have the plus there. All right, another one is insulin, frog versus human. Now, this requires perhaps a bit of information, although it's somewhat written out here. Insulin, actually, it is written here, but let me just rephrase. Insulin is generated as a single gene product, but in its mature process form, it is two sulfhydryl-linked proteins, two individual proteins, not very big, but two individual proteins. And it comes about from this kind of phenomenon. Wait one. Hi there. So, if you recall in various classes, labs, various things, we've talked about sulfhydryl-linked proteins. This is part of what it would be thought of as the quaternary structure of a protein, and it comes about through cysteine residues that have a reactive sulfur group, and you form what's known as a sulfur-sulfur bond, or a disulfide bond. And this disulfide bond is a covalent link. And so you end up with a protein that can be covalently linked to another protein. And they're linked using these cysteine disulfides. These disulfides actually can be broken apart by using something called a reducing reagent, or a reducing agent. Beta-mercaptoethanol is a classic. It smells really bad, so I don't use it very much. It smells a little bit like rotten eggs. It is the odorant put into natural gas, and it will break apart the disulfides. Now, the point is, insulin is generated as a single protein that would really look like this, except in the Golgi, this disulfide bond is formed, and this end is proteolytically cleaved. That is, there's a region in the middle of the protein that is going to be cleaved out. That's the region that's not going to have a whole lot of homology. Because since it's not in the mature protein, it's not subject to any kind of evolutionary pressure, and so it can change at a fairly fast rate. So, just as a little note, insulin is now produced for people that have diabetes as a recombinant DNA-based protein. It's called humulin. And back, prior to 1985, 86, insulin was purified from pigs, from the pancreases of pigs, and people would develop various allergic reactions and things to this, and it was not as pleasant. One of the very first genetically recombinant engineered products was this human insulin gene, and it's produced in bacteria. And I actually used to think, bacteria are not going to process this properly. How does that work? Well, it turns out that they don't process it, and humulin looks like this. It actually has the loop, but it doesn't affect the insulin binding to its insulin receptor, and so they don't bother trying to do any kind of post-processing. We'll start talking more about some of the aspects of biotechnology and some of the drawbacks to growing things up in bacteria because they don't process the way eukaryotes do, and we'll see some of the ways we can get around that. So that's what you're looking for. Do all the various tasks and then answer the questions. If you go into BLAST there is sort of a search procedure or information buttons, and it'll tell you a lot of these things. This is a way for you to explore BLAST on your own. BLAST is a critical tool for molecular biologists. We'll be doing more DNA sequence analysis with BLAST as we carry on, even carrying out some in vitro PCR using BLAST. And that's all I have to say about that. I'm looking at the sequence to see if there's anything else I need to tell you about. Oh well. That's what you're working on here. We're going to continue on moving into some of the genome organization chapters and kind of move through them a little bit more quickly to get into some ideas of biotechnology and also some of the getting into issues in genome evolution. All right, that's what I had to say about that. Turn this in, like I said, a printout and stapled and brought to me is fine. Good to see you guys, and I'll check in with you later. Bye.